Hello viewers, welcome to CEC Gurukul Live Lectures. This is a second lecture in the series on amino acids and proteins. In my first lecture, I had discussed what are amino acids. So amino acids are compounds which have the carboxylic group and the amino group in the same molecule. Then I had gone on to discuss the naming of amino acids, and we know that amino acids are named popularly by their common name, and we also know that each amino acid can be represented by a 3-letter abbreviation or a 1-letter symbol. For example, if I talk of glycine, which is the simplest amino acid, then the 3-letter abbreviation for glycine is GLY, and the 1-letter symbol is G. So continuing in the same vein, today I shall be completing the naming and the history of the rest of the amino acids. So next in the series, the amino acid is asparagine. Asparagine was isolated from asparagus juice by Perry Wien and Louis Nicolas Vauquelin in 1805. And its symbol is ASN, 3-letter abbreviation is ASN, and its symbol is N. And what we find is that this asparagine has a amido group in its side chain. Not only this, if we talk of the uses of asparagine, then it is a very important amino acid because it is used in biosynthesis of glycoproteins. It also stimulates the nervous system. It protects our liver, and what we find is that asparagus, soya, whole grains, chicken, beef, seafood and eggs are rich sources of asparagine. The next amino acid, which is glutamine, which can the 3-letter abbreviation for which is GLN and the 1-letter symbol is Q. This again has a amido group in its side chain, and it was isolated by Schulze from beet juice. Glutamine is the most abundant and active amino acid. It is mostly produced in muscles and legs. Now this amino acid is converted to glucose when our body does excessive exercise or when we undergo a lot of anabolic activities. This amino acid helps to maintain the blood sugar level, and it also maintains the proper pH range. It is also very vital for the proper functioning of the immune system. The next amino acid is lysine, which was isolated by Dreschel from casein, that is the milk protein. It is represented by the 3-letter word that is LYS, and its symbol is K. It has an amino group in its side chain. Now the lysine is an essential amino acid which is necessary as a building block for all proteins. It plays a very important role in calcium absorption, and it helps in building muscle protein. Not only this, it also produces hormones, enzymes and antibodies. Quinoa, pistol, soya bean, lentils and other beans and peas are rich sources of this amino acid. The next amino acid is arginine. Arginine is the 3-letter abbreviation for arginine is AR and ARG. And now if I talk of ARG, there is a sound of R in it. And this arginine then is hence represented by the symbol is R. It was isolated by Ernst Schulze in 1886 from lupin seedlings. This amino acid has a gonado group in its side chain, which we can see at the end, which is NH2 double bond NH2 positive and NH. Arginine plays a very important role in cell division and the immune functioning. It helps in immune functioning by healing the wounds. Not only this, it also releases hormones and removes ammonia from the body. It is an intermediate in the urea cycle, and it is cleaved into ornithine and urea in the urea cycle. Now what we find is that turkey, pork, chicken, pumpkin seeds, pea and dairy products all are rich in this amino acid. The next amino acid that is in the series is phenylalanine. Phenylalanine is a derivative of alanine amino acid, in which the hydrogen of alanine is replaced by a benzene ring, and its 3-letter abbreviation is PHE, and this PHE sounds like fur. So the 1-letter symbol for phenylalanine is F. It was isolated by Schulz and Barberry in 1879 from the seedlings of lupin plant. Phenylalanine is a precursor of amino acid tyrosine. Now this amino acid helps in the formation of adrenaline. Now since we know that adrenaline is an exciting hormone, so what happens is that we can also say that phenylalanine is responsible for promoting mental health, alertness and memory, and it also helps in elevating the mood. So that means this particular amino acid is an exciting amino acid because it helps in the formation of adrenaline, and if in the case of crisis or some happiness, we feel good because it helps in the formation of adrenaline. Not only this, it also suppresses our appetite. Now, another very important thing about this phenylalanine is that sometimes what we find is that the enzyme which is required to utilize phenylalanine is missing. Now, if this enzyme is not there, which will metabolize phenylalanine in the bloodstream, this phenylalanine keeps on accumulating in the blood, which leads to a very rare metabolic disorder, which is phenylketonuria. So that means phenylalanine should be metabolized, and if it is not metabolized, it is then leads to phenylketonuria, and this disease is also a genetic disorder, and people who have this disease should eat less of proteins and more of simple foods like carbohydrates. The next amino acid is tyrosine. Tyrosine, the 3-letter alphabet abbreviation for tyrosine is TYR, and it is represented by the word Y. It was isolated by von Liebig in 1846, and we know von Liebig is father of biochemistry. Tyrosine can be considered, as we've already said, to be derivative of phenylalanine, and when we talk of it being a derivative of phenylalanine, what we find is that the one of the H has been replaced by an OH. So what we say is that in tyrosine, the side chain has phenolic group in it. Now, the tyrosine represents a starting material for the neurotransmitters like dopamine and non-adrenaline. Non-adrenaline is very important for healthy brain functioning, and this particular amino acid, that is tyrosine, helps in relieving pain in the body, and it also prevents conditions like depression, because as I've already said that tyrosine is a starting material for neurotransmitters like dopamine and noradrenaline. So that means it is giving us some transmitters which will consider the pulses from one nerves to the other, and therefore it will even help in relieving the pain, and it will elevate our mood and thereby removing depression. Tyrosine is present in almonds, it is present in avocados, then it is present in chicken, milk products and bananas. The next amino acid is proline, and as we can see from the structure, proline does not have a amino group, it has a NH group, and this NH group, here we've said NH2 positive, which I've shown here that it is a amino group has been protonated. So it is a amino acid, and it was isolated by Willstatter in 1900, and this has a pyrrolidine structure in this amino acid. And what we find in proline is that its R group is linked back to its own amino group, as a result of which a ring structure is formed. And because of formation of this ring structure, we find that wherever this proline is present in protein molecule, it will cause bends or kinks in the amino acid chains because of its folding itself back on its own amino group. Proline is a non-essential amino acid. It can be obtained from glutamic acid, and it is an essential component of collagen. And since it is an important component of collagen, what we find is that it is very important for the proper functioning of joints and tendons. And since it is present in collagen, it also helps to maintain the strength of heart muscles. It can be easily utilized as an energy source by the body in case of the low amount of glucose or when the anabolic activity is more. The next amino acid is histidine, which is represented by the 3-letter alphabets, which are HIS, and its 1-letter symbol is H. It was discovered in 1896 by Albrecht Kossel and Sven Hedin independently. They discovered it separately, histidine. And what we find in histidine is that it has a imidazole ring in the side chain, and it is this imidazole ring is present in important biologically important compounds like histidine and histamines. Histidine is a semi-essential amino acid. What I mean by saying it's a semi-essential amino acid is that adults are able to produce an adequate amount of histidine, but however children cannot synthesize it or they cannot produce it. So when we talk of children, children need to be given diet which is rich in histidine. And as I've already said, that in histidine, the molecule that we have in histidine is the imidazole ring, and this imidazole ring, as I've already said, is present in histidine as well as histamines. So what we find is that this histidine is a precursor of histamine, and histamines are released by the immune system in response to an allergic reaction. And not only this, histidine is a very important amino acid because it helps to regulate the essential trace elements like iron, copper, molybdenum, zinc and manganese. That is what I mean here is that iron, copper, molybdenum, zinc and manganese, which are trace essential elements, they are utilized in presence of histidine. The next amino acid is tryptophan. Tryptophan was isolated from milk protein casein by Sir Frederick Goland Hopkins in 1901. And what we find that in tryptophan, the side chain has indole ring in it. Now, what is an indole ring? Indole ring has a 6-membered ring which is fused to a 5-membered ring, and indole is a heterocyclic aromatic compound, and this tryptophan has this indole ring in its side chain. Tryptophan is an essential amino acid since it cannot be manufactured in the body and it must be obtained from the diet. And it is a very important amino acid because it is needed for protein synthesis. It also helps with the production of the hormone serotonin, niacin, which is vitamin B6, and a plant hormone, which is auxin. Serotonin is a hormone that transmits signals between the nerve cells. So it is very important, tryptophan is very important again for the proper functioning of the nervous system, since it helps in the production of serotonin, which is an hormone which helps to transmit the signals between the nerve cells. Now, all these 20 amino acids are coded by the universal genetic code. And if we say that in between the genetic code, that when we are synthesizing polypeptides from these 20 amino acids, then in between the synthesis of polypeptides, if nonsense codons like UGA interferes, then what we find is that the protein synthesis or the polypeptide synthesis stops at that stage. And therefore, these nonsense codons are also known as stop codons. But however, it has been found that 2 amino acids can be synthesized in bacteria which are coded by this nonsense codon, among one of which is UGA. So what we can say is that nonsense codons become sense codons when we talk in terms of bacteria. So what we find is that if I talk, so now 2 more amino acids have been discovered in bacteria which are synthesized with the help of these nonsense codons, and these are the 21 and the 22nd amino acid. And if I talk of the 21st amino acid, it is celinocysteine. That is, it is a derivative of cysteine, in which if you look here, the S group of the thiol, that is SH group, has been replaced by a selenium group. So now when we name this compound, that is when we give the abbreviation, the 3-letter abbreviation for celinocysteine, so we take the 2 alphabets of the seleno, that is SE, and then we take C from cysteine, which indicates that this is a modification of cysteine molecule. And the 1-letter unit for is U. And then this celinocysteine is coded by the stop codon UGA, and it is present in several enzymes like glutathione peroxidases and some hydrogenases in bacteria. The 22nd amino acid is pyrolysine. As the name indicates, this pyrolysine is again a derivative of lysine. And when I talk of, and it was discovered by Kriski, and it is again, this pyrolysine is coded by the stop codon UGA, and its symbol is O, and the 3-letter abbreviation for this is PY, which comes from pyrrole and pyro, and the word L, which comes from lysine, which indicates that it is a derivative of lysine amino acid. And it is now present in methyl transferase enzyme of bacteria. So till now what I have done is I have discussed the history of amino acids, that is who isolated amino acid from where. We've also discussed the 3-letter abbreviation for amino acids and the 1-letter symbol for amino acid. Then we have also discussed the different uses or the functions of amino acids. Now we will proceed on to classification of amino acids. And when we talk of classification of amino acids can be classified on two bases. First is the nutritional. Now when I talk of nutritional, that means that we will be talking of essential amino acids. That is, essential amino acids are those amino acids which cannot be synthesized in the body, hence they have to be provided in their diet. And then there are other amino acids which are the non-essential amino acids which can be synthesized in the body. Second basis of classification is that we can talk of the classification on the basis of group R. As I've told you in my previous lecture also, that when we talk of amino acids, then we say amino acids are those compounds which have the amino group and the carboxylic group in them. So the basic unit for an amino acid is the amino group and the carboxylic group. So when we talk of classification of amino acids, as I've already said, that we can either classify them on the basis of nutritional level, or we can classify them on the basis of the group R. So when we talk of an amino acid, it has a basic group. Basic skeleton consists of the amino group and the COH group. And that means the classification will now be done on the basis of group R, which is present on the side chain. That means this group R can either be polar or it can be non-polar. That means this R group, or the side chain as we call it, is going to be different for each amino acid, and it is going to give its characteristic properties. That is, the structure may be different, the size of the R group may be different, its electric charge may be different, and when the electric charge will be different, it will affect the solubility of amino acids in water. That is, the amino acid may be hydrophilic or the amino acid may be hydrophobic. So depending on the polarity of the R group, we can classify amino acids into non-polar aliphatic amino acids, polar uncharged amino acids, aromatic amino acids, acidic amino acids, and basic amino acids. So now let us go back to the structure of the amino acid. So what I want to tell you here is that when we talk of a physiological pH, that is pH between 7.2 to 7.4, and as we've seen in amino acid, we have a COOH group as well as amino group. So that means in the same molecule there is an acidic group as well as amino group. So what happens here is that is an internal acid-base reaction takes place, as a result of which amino group acts as a base because of the presence of lone pair of electrons on nitrogen, and it is protonated. So that means it accepts a proton from carboxylic group and it has a positive charge on it. Whereas when we talk of a carboxylic group, it is acidic in nature and it is giving a proton to amino group. So hence it undergoes deprotonation and it is negatively charged. So what we find is that when we talk of physiological pH, that is pH between 7.2 to 7.4, amino acids are present as Zwitter ions. Now what I mean by saying it acts as a Zwitter ion, that is, it is electrically neutral ion and it is a non-polar ion. And that means now it is the group R which is bonded to the central carbon, which will now determine what is the identity of this amino acid, that is whether this amino acid is non-polar, polar, or it is an acidic or a basic amino acids. Let us look into some examples. That means that if we talk of valine and leucine, what we know is that valine and leucine have a hydrocarbon, aliphatic hydrocarbon side chain. So these are non-polar in nature. Now since they are non-polar in nature, they are going to be hydrophobic. And similarly, if we talk, on the other hand, if we talk of serine and glutamine, what we find is that these amino acids have polar side chains and they are hydrophilic in nature. That is, serine has a OH group in the side chain and glutamine has a amido, that is CONH2 group in the side chain. Similarly, if I talk of lysine and arginine, then what we know is that lysine has an amino group in the side chain and arginine has a guanidino group. And then because these have positively charged groups in their side chains, these are basic amino acids. Similarly, if we talk of aspartate and glutamate, they are also charged, but they are negatively charged because at physiological pH, they are acidically in nature. That is, they have one carboxylic group extra in them. Hence, they are acidic in nature. So now let us discuss non-polar amino acids in detail. So if I talk of non-polar amino acids, then what we know is that is the non-polar groups can be alkyl groups or they can be aromatic groups. So that means in the side chain, the group present will be either an alkyl group or it will be an aromatic group like a phenyl group. And these side chains of these proteins do not accept or give protons since they are non-polar in nature. And since they do not have a hydrogen attached to an electronegative atom, they do not form hydrogen bonds and neither are they capable of forming ionic bonds. Hence, we consider that the side chains of hydrophobic amino acids, or rather non-polar amino acids, are oily or they are lipid-like. Now, since they are oily or lipid-like, they are going to promote hydrophobic interactions, and therefore the interaction between these non-polar amino acids are going to be weak van der Waals forces, which will give stability to the protein molecule. Now, if I look of the aliphatic amino acids, that means which have an aliphatic side chain, then what we can say is the simplest amino acid is glycine, and in glycine the side chain has having H, and this is the simplest amino acid which is optically inactive. Now the next is alanine, in which the side chain is having a methyl group, and the next amino acid is valine. And when we talk of valine, I have already told you that it has an isopropyl group, and we can think of valine as having a group which is a inverted V. Then we have isoleucine, and if you look into the structure of isoleucine, we talk of leucine, then what we find is this leucine is in leucine, the group present here is a isobutyl group. And next amino acid is again methionine, which has S-methyl group on it. And the next one is isoleucine. Again, this, as I said, is a misnomer here because the group here is a secondary butyl group. So what we find is that whether we talk of a glycine, we talk of alanine, we talk of valine, leucine or isoleucine, these have aliphatic hydrocarbon chains in it. That is, they have alkyl side chains, and these alkyl side chains are non-polar in nature, and they promote hydrophobic interactions within the protein structures. If I talk of aromatic side chains, that is, the side chain has an aromatic group, then we can talk of phenylalanine, which has a benzene side chain, which, as I've already said, is considered to be a derivative of alanine. And then we have tyrosine. Now this tyrosine is considered to be derivative of phenylalanine, and it has a phenolic side chain. Now since it has a phenolic side chain, that is, it has a OH group in tyrosine molecule. So that means this molecule is capable of forming hydrogen bond, and it plays an important function in the activity of some enzymes. And the next aromatic chain that we have is tryptophan, and as I've said that in tryptophan the side chain is aromatic in nature, and it is a indole ring which is made up of a one ring in 6-membered ring is fused to a 5-membered ring. And again, these are again non-polar, and since they are relatively non-polar, they will again participate in hydrophobic interactions. Now if we talk of the next category of amino acids, then they will be polar uncharged amino acids. That is, they are polar in nature because of the presence of some polar groups like OH, NH2 or a thiol group, but they will neither be acidic or basic because they have equal number of amino and carboxylic group. That is, if it has a one amino group, it will have one carboxylic group, which, as I've already said, is the basic structure of an amino acid. And the side chains of these amino acids will contain different polar groups, like those polar groups can be amines, they can be alcohols, or they can be sulfur groups. And since they have these polar groups, these amino acids are hydrophilic in nature. Now when we talk of hydrophobic groups, they occur deep inside the protein molecule. Above them are the polar molecules, and above them then we have either the acidic or the basic amino acids. So now when I talk of polar uncharged R groups, then we talk of the first is the serine, and when we talk of serine has a hydroxyl group in it. Similarly, if I talk of threonine also has a OH group, that means it has an aliphatic group. So we can say that if I talk of serine, that serine is a derivative of alanine in which one H has been replaced by a OH group. And then we have tyrosine, which I have already said, tyrosine has a phenolic group in it. Cysteine has a thiol group in the side chain, and as we've already discussed that asparagine and glutamine have the amido groups in them. So that means these polar molecules are uncharged because the number of the carboxylic group and NH2 amino group are same. It is only that the side chains are polar because of the presence of the hydroxyl group, amido group or a thiol group. And since they are polar in nature, they are more soluble in water as compared to non-polar amino acids because they are capable of forming hydrogen bonds with water. Now, if I talk of cysteine, then what we know is that cysteine contains a thiol group, which is SH group, and it can form covalent bond with other cysteine molecules. So now if we do the reaction between two cysteine molecules, then what we find is that these cysteine molecules undergo oxidation, and that means during oxidation the two hydrogens are removed and a covalent disulfide bond is formed between the sulfur atoms of the two cysteine molecules, resulting in the formation of cysteine. And this cysteine then plays a very important role in enzyme and protein activities. The third category of amino acids are the acidic amino acids. So now if they're acidic in nature, what we have to remember is that the side chain has to have an extra component, that is, it will have a carboxylic group. And since it has a carboxylic group, it will have the tendency to give a proton molecule. It will give a proton atom or an hydrogen atom. And since it is giving a proton, that means this acidic amino acids will have a net negative charge on it. And that means they will have net negative charge, as I've already said, at the pH 7. Now, if we talk of basic amino acids, then what you have to remember is that as in acidic amino acids had one carboxylic group more in the side chain, the basic amino acids will have one basic group or amino group in the side chain. And now if I talk of lysine, then in lysine the basic group is the amino group, that is the R group here is the amino group which is making lysine basic in nature. And now if we talk of arginine, then in arginine the basic group or the side chain has gonadino group. So gonadino group is NH single bond C double bond NH2 positive and then a single bond NH2. And this is the basic structure of gonadine. In gonadine what we have is C, NH2, double bond NH2 positive and NH2. So this molecule is arginine is basic in nature because of the presence of this gonadino group in it. Now the last amino acid which is basic in nature is histidine. Histidine has an imidazole moiety in it, and these amino acids, basic amino acids, are groups which have a positive charge on it, because these basic groups will accept a proton and they will become positively charged, and we say these amino acids which have a basic group like a amino group or a gonadino group or a imidazole ring in their side chain are basic in nature. The last category of amino acids are amino acids which are aromatic amino acids. Please remember, we can put these aromatic amino acids in the non-polar series amino acids also, and we can discuss them separately as aromatic amino acids also. And these are three, which are phenylalanine, tyrosine and tryptophan. So now when I talk of phenylalanine and tyrosine, these are the groups, these are the amino acids which have a benzene ring in them. Whereas when we talk of tryptophan, then tryptophan has a indole ring in it, and it is a heterocyclic aromatic amino acid. Now what I have done till now is I have discussed the amino acids in detail. That is, we know that we have 9 amino acids which are non-polar, we have 6 amino acids which are polar, then we have 3 amino acids which are basic in nature, 2 amino acids which are acidic in nature, and then we have amino acids which are aromatic in nature. Now the question arises, how are we going to remember these amino acids? It's very difficult to remember. As such, the acidic series, the polar series, non-polar series. So the easy way to remember the different sets of classes of amino acids is by making simple sentences to memorize them, and which we call them as mnemonics, which will help us in memorizing these series. For example, if I talk of non-polar amino acids, I would like to remember a sentence which says Glinda always visits London in May for trying probiotics. Now please remember, I have underlined the first alphabet of each word by writing it in red color, and that red color will indicate the name of the amino acid. For example, G stands for glycine in Glinda, A in always stands for alanine, V in visit stands for valine, L in London stands for leucine, I in stands for isoleucine, M for methionine, F for phenyl, Tri for tryptophan, and P for proline. So by remembering the sentence, Glinda always visits London in May for trying probiotics, we can remember the non-polar amino acids. Now, if we have to remember polar amino acids, how do we go about it? Again, please remember the first alphabet of the word, which is written in red and in capital, will represent the first word of the amino acid. That is, sages treat cyst with glucose and asparagus twigs. So, if you remember, see in this sentence, I have not written with W of with capital and A of and with capital. That means they are not going to indicate the amino acids. So if I talk of as sages, that means serine, treat means threonine, cyst means cystine, G for in glucose means glutamine, A means in asparagus means asparagine, and T means tyrosine. That means the polar amino acids we can remember by remembering the sentence, sages treat cyst with glucose and asparagus twigs. That means serine, threonine, cystine, glutamine, asparagine, and tyrosine. Now, if we talk of acidic amino acids, then we know all is good. So, all will stand for aspartate and G stands for glutamate. So, by simply remembering the sentence all is good, we know that we have two acidic amino acids, which are A for aspartate and G for glutamate. Now, if I talk of basic amino acids, then basic amino acids are high and low. In this, I have indicated the first alphabet of each letter with a capital. That means H will also indicate amino acid as well as A and L. So H is for histidine, A is for arginine, and L is for lysine. So high and low are representing which amino acids? They are representing basic amino acids. Now if we go on to aromatic amino acids, then you know that the world is for the youth. So what I have done is, I have written this sentence, or for world youth. In this case, what I have done is, I have not taken the 3-letter abbreviation for indicating the amino acids. Rather, I have taken the first word. So when we talk of for, that means for is indicating the symbol F for phenylalanine, W symbolizes tryptophan, and youth, Y of youth indicates tyrosine. So this is a simple method which I have proposed for you to remember the acidic, basic, aromatic, non-polar and polar amino acids. Because what happens is, sometimes in objective or in otherwise question also, they can ask us, name 2 aromatic amino acids or name the basic amino acids. So if you remember these sentences, then you know the first alphabet of the words which are forming the sentences will indicate or will tell us the either the symbol or the first word of that particular amino acid. So now next I would like to discuss is the non-standard amino acids. What are non-standard amino acids? Non-standard amino acids are found in small quantities in proteins. Now, these non-standard amino acids do not take part in protein synthesis. They are non-protogenic, and the two examples of these can be 4-methyl hydroxyproline and 6N-methyl lysine. That is, they are a derivative of proline. 4-hydroxyproline is a derivative of proline amino acid, in which we've replaced the OH on the 4th carbon. And when I talk of lysine, then what we find is that a methyl group has been put on the 6th amino group, and therefore we call it as the amino group which is at the 6th position, and it is known as 6N-methyl lysine. And both of them are present. This is present, the first one, hydroxyproline, is present in collagen, and the second one is present in myosin. The second non-standard protein amino acids are derivatives of amino acids. They do not again take part in protein synthesis, but they play a very important role in amino acid metabolism. For example, if I talk, then we can say that ornithine and citrulline will occur as intermediates in urea cycle in animals. So we've talked till now of the standard and the non-standard amino acids, and we've also seen how will we memorize the amino acids, the different classes of amino acids. Now, another way in which the amino acids can be classified, now if I talk from the biochemical point of view or totally from the medical point of view, then we say that amino acids can also be classified on the basis of the metabolic fate. What I mean by is, on the basis of the metabolic fate is to what are they going to be converted into. So on basis of this, the amino acids can be of 3 types. They can be glucoketogenic. What I mean here is they can be glucoketogenic, that is, they can be converted into glucose or ketones in our bodies. And the examples of glucoketogenic amino acids are isoleucine, tyrosine, phenylalanine and tryptophan. And then they can be ketogenic. Ketogenic means these are the 2 amino acids, that is leucine and lysine, which can be converted into ketones in our body, and the remaining 40 amino acids are glucogenic amino acids. So that means now we've done the classification on 3 bases. First is on the nutritional basis, which I'll be discussing right away, the essential and the non-essential amino acids. So we've discussed in detail the classification on the basis of the side chain, that is R, and also on the basis of the what is going to be the metabolic fate of amino acids in our body. So now if I talk of classification on the basis of nutritional requirements, then what I've already told you is that there are certain amino acids which our body cannot synthesize. Such amino acids are known as essential amino acids. That is, these are the amino acids which have to be provided in the diet. And then there are non-essential amino acids. These are the amino acids which our body can synthesize from some other precursors. As I told you, that tyrosine can be made from phenylalanine. So similarly, these amino acids can be made from one of the precursors. So let us see what are the essential or the non-essential amino acids. So if you remember, non-essential amino acids are the ones which are not required in the diet, and that means can be formed in the body. So they are alanine, asparagine, aspartate, glutamate, glycine, proline, serine, cysteine and lysine. And when I talk of cysteine and lysine, cysteine and lysine are non-essential amino acids which are synthesized from the essential amino acids, which are methionine. Now if I talk of the essential amino acids, then essential amino acids are the amino acids which our body cannot make. therefore what we have to do is we have to take them in diet, because if we do not take them in diet, they will lead to some form of deficiency, and these are arginine, histidine, isoleucine, leucine, valine, lysine, methionine, threonine, phenylalanine and tryptophan. Now here what we've done is we've shown lysine and histidine in red color with an asterisk. So what this indicates is that arginine and histidine are semi-essential amino acids. That is, children cannot synthesize these amino acids, but adults can. So these amino acids have to be fitted in the diet of children. And if these amino acids are not given in the diet, they may lead to deficiency of these amino acids, which may lead to different diseases in human beings. Now again, as I've done the mnemonics for the amino acids, the acidic amino acids, basic amino acids, I would also like to do the mnemonics for the essential amino acids. So we have PVT, private Tim Hall. So in PVT, each word is indicating a amino acid. So P is indicating phenylalanine, V is valine, T is threonine. If I talk of Tim, T is indicating tryptophan, I is indicating isoleucine, M is indicating methionine. Similarly, H in Hall is histidine, A is arginine, L is leucine, and L is lysine. With this, I finish with the classification of the amino acids. I hope this will be useful to you, and you also know how to memorize the amino acids, the different classes of amino acids, with the help of mnemonics. Thank you.